Hey everyone, this is chapter 15 of Essentials of Biology textbook. The title is Evolution on a Small Scale. So last chapter we essentially talked about the basic foundations of evolution and several different hypotheses that existed that several different people had come up with and the reasons behind sticking with Darwin's mechanisms behind evolution and his hypotheses. So this chapter, we're just going to expand on those ideas and talk even more specifically about natural selection and microevolution. So in the beginning of this chapter, we start off in 15.1 discussing natural selection, which is a process that we touched on pretty well in chapter 14, but now we're going to expand on it even more. So natural selection, as we discussed before, is a process that results in adaptations of a population to the living and non-living aspects of their environment. Living things being considered the biotic factors. You can see the prefix bio here, and abiotic would be the opposite, the non-living factors. So some living or biotic factors could be the interactions of predator and prey or other living things in the environment surrounding a population. The non-living things could be like mountain ranges or climate or weather or the grasslands, etc. It's the non-living aspects of a population's environment. And just for the record, let's go back to what a population is, because we're going to talk about populations a lot during the next chapter, or this chapter and the next. And if you recall, a population is a group of organisms of the same species. That's important there. They're the same species that breed and interact together. Okay, so this process of natural selection where a population adapts to the living and non-living aspects of environment was Darwin's mechanisms for evolution. In his ideals, the most fit individuals become more prevalent in a population, and that's because the individuals that have the most fit genes that then therefore have the most fit traits are more likely to survive and more likely to reproduce than those that don't. And thus, the gene pool, which is the term we'll discuss in a few minutes, is going to be more inclusive of alleles or genes that have more favorable traits. So this essentially leads to a change in the population over time. And the most fit individuals reproduce more than others because they're better adapted to their environment, and thus the next generation will have more prevalence of the characteristics that made the previous generation more fit. So for example, let's say for hypothetical all intents and purposes, if in this day and age right now it was more fit for people to have blonde hair versus brown hair or red hair or black hair, if the most fit gene for our environment was to have blonde hair, then less people would survive that had any other hair color, and therefore they would not be able to reproduce and pass their genes on. So in the next generation, we would see a lot more individuals that had blonde hair. Okay, so next, evolution by natural selection involves three things. The first thing is we have to have variation. If we all had, like we'll stick with my hair color example, if we all had blonde hair or if we all have brown hair, if there's no variation, then our populations would not be able to adapt. So variation is a requirement of natural selection. So the members of a population have to differ from one another. Secondly, another requirement for evolution by natural selection to occur is inheritance. So our physical traits that make us more fit or less fit for our given environment has to be something that's heritable, that can be passed down from generation to generation. And lastly, increased fitness. So the individuals that have the most fit traits are going to be more likely to reproduce, and their fertile offspring will make up a greater proportion of the next generation. So those are the things that are involved in natural selection. Now, something we like to do as scientists is to graph things. We like to graph data because when we have lots of data, it makes it so much easier to interpret our results if we can graph them. You can imagine if we were to take the height of everyone in all of my classes, and we were going to compare them to the height of all the students in all of the math classes. That would be a lot of numerical data, and it would be a pain to try to analyze or draw any conclusions from it by looking at a whole bunch of numbers. So graphing things is a really great tool across the sciences, as well as any other discipline, truly, to help us interpret large amounts of data and make sound conclusions from it. So when talking about physical characteristics in species, most of our traits are what we call polygenic. And I believe we touched on this term maybe in chapter like 9 or 10 when we discussed inheritance, and I think it's when we discussed inheritance. Anyway, we're going to bring that term up again, polygenic. If we break it down, we have poly and gen. Poly means many, genic meaning genes. So most traits are polygenes, or polygenic, meaning many genes. That means that the trait is produced by multiple genes. So a great example would be eye color. So when we talked about inheritance patterns several chapters back, sometimes I would discuss eye color as if it was a monogenic trait, like if it was only controlled by one gene, which means 2 alleles, right? Because you get one from mom, one from dad. So that means, we would we would have a blue-eye gene and a brown-eye gene. Those are only 2 options. So that would be a monogenic example. But in reality, even though sometimes as scientists will simplify things for students like that, eye color is really a polygenic trait. That's why we have, not just blue eyes and brown eyes. We also have green eyes, and we have different variations of green and blue and brown and mixes sometimes. So anyway, most traits are polygenic, having many genes that control that trait. A great example besides eye color is skin color and height. Because again, if it was a monogenic trait, then it'd be more likely to have just you either have this height or this height. You're either tall or short. You're either going to have very pale skin or very dark skin, but that's obviously not how the world works. In those types of traits that are polygenic, we consider those traits continuous, meaning there is a continual curve we can create with data and graphing that shows that the data is not just either or. So oppositely, a monogenic trait would produce a discontinuous set of data, and we would graph it like this with a bar graph. Because let's say, for example, we could have had brown eyes here and blue eyes there. Another trait we talked about in that chapter that I was referring to is some people have a widow's peak and some people have a flat hairline. So if have a widow's peak, you would be in this category. If you have the flat hairline, you would be that one. It's a hairline is a monogenic trait, which produces discontinuous data. So oppositely, the polygenic traits that we're discussing now that are controlled by many genes are going to produce a continuous set of data. It's not a scenario where you either have very light skin or very dark skin. There's a lot of different shades of skin tone, just like there's lots of different shades or different sizes of people, tall and short and everything in between. There's not just one of 2 things that you can fit into. So you would see a similar data set being graphed here if it was height. This one is skin pigmentation. So anyway, these curves are created by essentially taking a line and drawing it over the top here. Now, you can see it kind of produces a bell shape, right? So we have the big bell shape. And what this is showing you is that at the very top of the bell, most people are going to fall into this category. This is the average, okay? And then you're going to have a little bit less people fall into these two pigmentations. And then a little bit less fall into the next one, and a little bit less on the next one, and less and less and less and less, until we get very, very few people have really, really dark pigmentation, and very, very, very few people have really, really light pigmentation. Okay, so this is called a histogram, if I'm not mistaken. But once it's you create the bell curve from it, it'll look something like this. Okay, so this shows you the frequency of the phenotype, and this shows you the phenotype on the x-axis. So this one specifically is for height. So this would, be, let's say, 5 foot, and this would be 8 foot. All right, so most people would be, somewhere in the center, and relatively few people are over here, relatively few people over there, and everyone else kind of falls in between. Okay. Oh, one more thing I forgot to mention. This bell curve can move according to environmental changes. Okay, so let's say for all intents and purposes, all of a sudden something happened in our environment, and now it was really beneficial for us to be short on this end of the bell curve. We have a new prey, a new predator that is preying on humans, and they are able to easily see tall people versus short people. And now short people are being selected for. That's going to cause this curve to shift over a couple of generations, because we're going to see most of the tall people get eaten by the predator, so they no longer exist, and our curve will shift towards the left, where then the new average will be somewhere, over here. We'll have a new bell curve. I know my drawings are not the best, but I'll show you some more examples. All right, so what I was just referring to is a type of selection called directional selection. And this occurs where an extreme phenotype is favored. If, again, in my scenario, if really short people were selected for, it was a fit trait for our environment. Or let's say something happened in the environment, and now it was beneficial for fish to be able to survive cold temperatures, because the earth, all of a sudden, we hit ice age, something like that. Or if it was more beneficial for plants to be able to tolerate more heat because our earth is warming up, which is actually what's really happening. So that's a better example. But then that would mean that the plants that couldn't withstand warmer temperatures would die, and thus the next generation, we would see more of plants that were able to withstand that high heat. Thus, our bell curve would shift. All right, so the distribution curve or bell curve shifts in that direction. So this can occur when a population is adapting to a changing environment. Some examples: industrial melanism. We'll talk about that one more later on in the PowerPoint, so just tuck that away in the back of your head. Drug resistance and bacteria. We'll see a population of bacteria, and if you take an antibiotic and wipe out most of the bacteria, there was going to be some of them that are resistant that are going to survive, and therefore their offspring are going to also have those resistant genes as well. Shift in the curve. Pesticide resistance in insects. Malaria. Malaria is a disease caused by a plasmodium, which is a protist. It's a blood infection, and it's becoming more resistant to chloroquine, which is what is the standard treatment right now. And mosquitoes are becoming more resistant to DDT. Another example of directional selection is equus, which is the species name for horse, is adapting to forest conditions, or did adapt from forest conditions to grassland conditions. Because if we know about horses now, they are grassland type animals. They feed on the grasslands. Well, in their evolutionary history, is documented that they were long ago more of a forest type animal, like a deer, for example. So this is an illustration of the distribution curve movement over time for horse species. So again, this is directional selection. If we look back in their evolutionary history, long, long ago, this is the initial distribution, and this is related to body size. So you'll see the number of individuals is on the y-axis, and the body size is on the x-axis. So here we have really small horses or past horses. Their common or their ancestors, we'll say. And then we have the ones that had a very large body size, and then this is everyone else. Most of the horse or ancient horses were around this size. Well, as they migrated or became more adapted from forest to grassland habitats, it became more beneficial for them to have a larger body size. So after generations, many generations, what we're going to see is this curve shifted. So here we see it's all the way to the left, right? Now we're seeing it kind of in the middle. So after time, what we saw, because a larger body size was selected for and the smaller ones did not survive and reproduce, we see a shift in our bell curve, where now our average is here. Then after more time passed by, that curve shifted even further because the ones with the largest body size were the most fit, they produced the most offspring, and thus the tall genes were passed on to the next generation in high numbers compared to the ones that were small that did not survive and reproduce. So again, we see another shift in our bell curve over more time. Okay, so that is directional selection, right? The curve is shifting in a particular direction. The next type of selection is stabilizing selection. So in stabilizing selection, an intermediate phenotype is favored. The average is favored. And the extreme phenotypes are selected against. So the extreme phenotypes are not fit. This is the most common form of selection because the average individual is well adapted to its environment. So let's look at some examples. Here is a bird. These are called Swiss starlings. They lay around 4 or 5 eggs because this number has the highest survival rate for their young. If their clutch size is smaller or larger than 4 to 5 eggs, then those offspring tend to not do well, and the parents they don't survive as and continue to reproduce as frequently as those that only have 4 to 5 eggs. Okay, so what you'll see here's our initial distribution. Many generations ago. This is survival of the young and clutch size. So if there was less than 4 eggs, very few young survived and reproduced. Likewise, if there was more than 5 eggs, then relatively few of those young would survive and reproduce. So the birds that laid a clutch size around 4 to 5, you can see your key over here, by the way, 4 to 5 eggs were more likely to survive and reproduce. And so what you see is there is a narrowing of our bell curve over time. It narrows the distribution curve. Another example is human birth weight. So most babies that are born are going to be born of around, if we were to have a line be drawn down, around, let's say around 3 kilograms. So that's, around 7 point something odd pounds. So relatively few babies are born on this end of the spectrum. Relatively few babies are born on this end of the spectrum. So this is, let's see, the conversion factor for pounds to kilograms is 2.54. So for every if you were to multiply, this was around what, 4.4 times, you can multiply 4.4 times 2.54, and that would give you that in pounds if you would like that. If you would like to do the math there, if you're more comfortable with pounds, you can do that conversion. But babies essentially that are born that are very large and are very small are less common. And babies are born around, are most commonly born around 7-ish pounds, which is around 3 kilograms. And then you can see everything else fits in between. Now, if I want us to now take a look at this red line here. This red line is representing the percent of infant mortality for each weight. So we can see we have a relatively really high infant mortality, which is infant death, in this area that coincides with these weights. Let's say 7 pounds to, or excuse me, 0.7 kilograms to 1.7 kilograms. All right, so relatively pretty high death rates. As the babies get smaller and smaller, we see really high infant mortality. Likewise, though, everyone, that I know thinks, oh, it's so great. I have such a big baby. It's really healthy. Whenever they're pregnant, they have like a 10-pound baby or something. For some reason, society tends to think, oh, well, that's good. That means the baby's really healthy. And I'm not saying that you can't have very large babies that aren't healthy, but it is more likely to, for an infant to die whenever it is very large. When it's very large and when it's very small. And it's more likely for an infant to survive. There's less infant mortality. The least infant mortality is right here, right where most babies are born, in that average range, around 7 pounds. So now I'm not trying to scare anyone by any means. Infant mortality is really not common, but there is data that shows that offspring that are in the average for birth weight are more likely to survive. So, for example, my baby, when he was born, was 5 pounds and 10 ounces. He was a very small baby, and he's totally healthy. He's 2.5 now, so again, I'm not trying to freak anyone out, just to educate you. Okay, so again, this is stabilizing selection. Stabilizing selection is where the average is selected for. It's the most fit. And I mean, imagine why. Why do you think that is? Why is it beneficial for babies to be an average size, like around 7 pounds? Well, these infants that are really, really large, think 10-pound babies or more, they typically are not able to be born naturally. They have to have, the mother has to have a C-section. Excuse me. Now, previously, to modern times, if a woman needed a C-section, it was more risky. Still, typically, C-sections can be more risky than natural births. Think even further back. There was a time where, C-sections, of course, were not a thing. So if there was very, very large babies, they would not be able to survive the birth. And oftentimes, the woman would not survive that birth either. Further corroborating the stabilizing selection. Okay, the third type of selection is disruptive selection. This is where there's 2 or more extreme phenotypes that are favored over the intermediate. So what's going to happen is over time, our single initial distribution curve is going to start separating into 2 separate curves. So a great example of this would be in British land snails. Those that live in the fields, birds, like the birds that eat them are called thrushes. They eat the snails with the dark shells that lack bands, like these guys. But in the forest, those thrushes feed mainly on snails with the light banded shells, like this. So what we would see over time is a disappearance of anything that was in between. So we would have 2 separate curves. All right, so all in all, these are the 3 types of natural selection that we discussed. So the first one was directional selection. This is where we have a bell curve that's going to move in a particular direction, left or right, depending on the environment. If this in blue is the area that's being selected, is the where is the data for the organisms that are being selected for, we'll see the curve move to the left. Or excuse me, I'm sorry, to the right, that way. All right, and this is the result. So this was our initial peak here, and now we saw it move to the right. Secondly, we discussed stabilizing selection. This is where the average is selected for, where we are selecting for the organisms in blue. These are the ones that had the most fit genes. These don't. So what we're going to see is a narrowing of the peak. So you can see the original one is in the dotted line, and we see a narrowing of our distribution curve. And then lastly, our disruptive selection. So think of it like this: we're disrupting that peak. These are the organisms that are not going to have the ideal traits, and these are. So we're going to see after time, this distribution curve is going to separate into two separate peaks because the intermediate phenotype that was here died. Okay, so that was natural selection. It happens naturally. Another type of selection we need to discuss is sexual selection. So what sexual selection is are adaptive changes in males or females that lead to increased ability to secure a mate. Typically, in animal populations, males are going to compete for their females, and the females choose the males that they mate with based on which ones are the most fit. And fit in the animal kingdom, when we're discussing sexual selection, can mean different things for different species. But all in all, no matter which species population we're talking about, the female will choose the one that they deem the most fit, quote unquote, to mate with. And fit can mean the biggest, or the most colorful, or the one that has the biggest antlers. So for example, in peacocks, the females are attracted to all of these eyes on their feathers. So it'd be the peahen is attracted because you have the peacock and you have the peahen. The hen is the female. So she would be attracted to all these beautiful eyes on the plumage of the peacock. And the ones that have the most eyes are the most attractive to the females because their instincts tell them that the more eyes they have on their plumage and the more beautiful that bird is, the more colorful it is, then it must have the best genes. It's a sign of health and vigor for that bird. Just like in other populations. So these are cardinals here. This is the female. They tend to, in bird populations, the females tend to be like the drab ones that aren't very pretty. And it makes sense as to why, because the females are the ones that lay the eggs and they're trying to have to protect their eggs. So it's beneficial for the females to be the drab, dreary, dark, not pretty colors, because they need to blend in with their environment. So they're typically not going to have those really bright, beautiful plumage that you see in the males. This one over here is then therefore the male cardinal. And so you can see he has the bright red, so beautiful, and the female is very attracted to that because that is a sign, sure, her instincts that tells her that bird that has all those pretty feathers is very healthy and is very vigorous and very strong male, and therefore will give her strong offspring. All right, and so other animals like these here, or even the white-tailed deer that is really common in our area, the females are going to mate with the ones that they feel like are that have the best characteristics for male competition. So these males physically fight for those females, and you can see that's what these antlers, of course, are used for. And the ones that win the fight are going to mate with the females. Then the other ones just have to find another female or wait till next year. All right, so all in all, though, these adaptations are not perfect. Natural selection does not produce perfectly adapted organisms. Evolution as a whole is constrained by available variation. So again, we evolution is constrained by available variations. If there's not individuals in the population that have a particular trait, then that trait is not just going to, pop out of nowhere. Remember, this is a natural process that is just, it's an organisms are selected for based on the traits that they have. There is no like design to there's no design to or thought process behind it. Like I can't think, it'd be really beneficial if my muscles were bigger, and it just happened. And it wouldn't be that my genes would change. We're constricted to what we have. And the imperfections are common because of necessary compromises in our evolutionary history. So think about this, the success of humans, how well adapted we are to our environment, and how we're, essentially one of the most, if not the most, adapted species. But our ability to walk upright, which is beneficial to us because it frees up our hands and allows us to, do things, carry stuff, gather things. Nowadays in modern times, it enables us to drive and cook. I mean, think of all the things that we use our hands for. If we were walking on all fours, then it would be much more difficult to do that. But as a compromise, that upright position puts stress on our spine. So as we age, people tend to have back problems, slip discs, and just, arthritis. We can get scoliosis. And there's several other spine issues that can present themselves with old age. So, but it was a compromise that was necessary. It's not we're not perfect beings. We had to work with what we got or what we had. Okay, maintenance of variation. So a population always shows genotypic variations. And populations with limited variation may not be able to adapt to a changing environmental conditions very well. Think of it like this. Let's say that we have a culture of bacteria that are growing on a nutrient agar that contains glucose. Well, let's say I want to continue a line of growing that bacteria, but I'm out of the nutrient agar that contains glucose, and all I have is nutrient agar that contains sucrose. Well, if I take some of those bacteria and put them onto my new plate, which just has sucrose in it, the bacteria that can't digest that sucrose is going to die, and the ones that will live. There's variation in the initial population if that's the case. Some of them could digest sucrose and some can't. So those that could had more favorable traits for the particular environment that I put them in, which would be the nutrient bed that had the sucrose. So forces promoting variation are constantly at work. So some examples of promotion of variation includes random mutations. Remember we talked about DNA polymerase, what seems like a long time ago, and how its function was to essentially bring in new nucleotides while creating new strands of DNA. He's not perfect and he makes mistakes. And sometimes those mutations, those changes in the DNA that occur can be detrimental to the organism. Sometimes it might not have any effect at all, and sometimes it can be beneficial. So if it's beneficial, then it will be promoted in the population. Recombination, that's another example of promoting variation that we discussed in previous chapters, where non-sister chromatids would exchange components and then be distributed into the gametes. Independent assortment promoted genetic variation. That was where our homologous pairs would line up independently of all of the other ones. It would be at random. Fertilization itself creates new combinations of genetic material. Some other examples of forces promoting variation is gene flow. We'll talk about what that is in a minute. But natural selection favors certain phenotypes, but others remain. And the fact that others remain besides the favored phenotype is explained by diploidy and heterozygotes and the heterozygote advantage, which we'll talk about next. But diploidy, let's recall what that means. Diploids means that for every chromosome, you have two of them, one from mom and two from dad. And so because of that, organisms could be homozygous recessive, homozygous dominant, or heterozygotes. So let's relate that to heterozygote advantage. All right, so only alleles that are expressed are subject to natural selection. What does that mean? If an allele is expressed, then it is visualized, it's seen. So for example, let's say, and we'll go to eye color, pretending it's a monogenic trait again. So if big E is brown eyes and little e is blue eyes, and let's say that my genotype for eye color is big E, little e, then that makes me heterozygous, correct? Because I have one of each allele type. But which color will my eyes be, blue or brown? They'll be brown because in this scenario, big E, little e, the dominant trait or the dominant allele will mask that of the recessive. So my eyes would be brown. So again, the alleles that are expressed are the ones that are subject to natural selection because the ones that are expressed causes the phenotypic differences. So heterozygotes can protect recessive alleles. All right, because that means that even if you have a little e, you'd still be able to pass it down to the next generation, and it would not affect your ability to survive or reproduce because you express the dominant phenotype. Now, that's beneficial to a population because the recessive allele might have greater fitness in a changing environment. So right now, let's say brown eyes are the fit trait. Well, 3 generations from now, maybe blue eyes will be the most fit trait. And truly, eye color is not like a great example of truly fitness because no one's going to die because they have brown eyes or blue eyes. But think about it like this. Let's change it from eye color to fur color of mice. So I think we might have given this example before, but let's say we have a population of brown mice and instead of blue, let's put black mice. Well, if these mice lived in a area that is very rocky, the ground is very dark, then these mice that have black fur are going to be selected for. So we're going to see a lot more organisms over time that have the genotype little e, little e. We'd still see some of the others, but it would be most common to have the black fur color over time. Now, let's say that a sandstorm rolled in, and now the ground is brown. Well, then we would see a shift in our genotype or allele frequencies. We'll talk about what that means in a few minutes. Back to being, big E, or big E, little e, because the dominant allele would be selected for in that scenario. But this heterozygote protects that little e. It protects that recessive allele. It keeps it in the population because it might be beneficial in a changing environment. Maybe things will go back. All right, so that brings us to the term balanced polymorphism. Balanced polymorphism is when natural selection favors the ratio of 2 or more phenotypes in generation after generation. A great example of this is sickle cell disease. So I know y'all had an assignment on sickle cell disease in lab. So what's we'll reiterate some of the points about it. Sickle cell disease is a blood disorder where the hemoglobin gene, which is the gene for the hemoglobin protein, is mutated. And that mutation of the hemoglobin gene results in the hemoglobin protein being misshapen. And as a result of that misshapen hemoglobin protein, it has a domino effect on the overall shape and characteristics of the entire red blood cell where hemoglobin is found. So in normal individuals, red blood cells have this like biconcave shape, and they're very pliable and elastic cells. They can, kind of morph shape and squeeze through really tiny blood vessels in our body. And that's really good because we need our blood cells to be able to be moved throughout our body so these red blood cells can deposit oxygen in our tissues, which need it to survive. So in the case of someone who has sickle cell disease, their hemoglobin proteins are misshapen, and it causes their entire red blood cells to have kind of like a sickled shape. It looks kind of like a banana. And these sickle-shaped cells are not pliable. They're very rigid, and they get stuck and trapped inside of the really tiny capillary beds, which are really small blood vessels throughout the body. And so that creates an anemia in the individual where they have a decreased oxygen carrying capacity because these red blood cells get stuck and then they get destroyed. And that decreases the number of red cells as well as the ability to carry oxygen. So that's what sickle cell disease is, and it's a result of a single nucleotide polymorphism in the genome. Now, individuals with sickle cell disease, their genotype is as follows: HB, S, HBS. So that HB stands for hemoglobin, and the S stands for sickle. These individuals that are homozygous for this gene, HBS, HBS, they tend to die early in life because those sickle cell genes that produce sickle cells in the blood tend to create medical problems. This is especially common for them to die in third world countries. Here in America, there are treatments for sickle cell disease. Typically, these patients have, and I've witnessed it, it's pretty terrible to watch, but these people that come into the hospital with a sickle cell crisis, they're tired, they're in a lot of pain, because you got to think all these little sickle cells are getting trapped in various capillary beds in their body, and it's basically, kind of creating almost like mini clots sort of throughout their body, and that's extremely painful because they're not getting oxygen to their tissues. So anyway, there's lots of other symptoms there that we could go on and on, but I'll save it. But essentially they have medical problems and tend to die early relative to a heterozygous organism, or of course the normal genotype. Our heterozygotes for sickle cell disease carry the sickle cell gene, HBS, but they also have a copy of the normal allele, HBA. Their red cells only sickle at low oxygen concentration. So as long as they're not under excessive pressure or their blood cells aren't stressed out, essentially, they're fine. They can have this sickle cell trait they don't have the disease. They have the trait that they carry. A person that just has sickle cell trait can live their whole life without knowing it potentially, but definitely not the case for those that have sickle cell disease. Someone that has a normal genotype for the hemoglobin protein are the most fit, typically seen anyway as the most fit. They have HbA, HbA. The recessive allele HbS, however, has a higher frequency in regions in Africa, where malaria is present more present there in pretty much anywhere else in the world. And malaria, as if you'll recall from the beginning, is a parasite. It's a protoparasite that invades red blood cells. It destroys them and is ultimately something that can be fatal if left untreated. And it's a pain to treat too. If you've ever gone out of the country, you know all the preventative methods to prevent you from catching it. It's a lot of I can't remember if they have injections now, but they used to be really huge pills. I think it might be a combination, but I've never had to get them, so I'm not really sure. But I have heard from people that I know that have traveled outside of the United States that it is not fun. But anyway, so individuals that have this sickle cell gene are more common in populations in Africa where malaria is present. Why? That's so odd, right? Well, there's a reason. And it's, again, heterozygote advantage. Parasites are unable to live in a heterozygote red blood cell. So, because they are a heterozygote for this condition, it basically safeguards them from contracting malaria. But we see we have a compromise here. They have sickle cell trait, or of course sickle cell disease. People would not be able to get malaria as well. But of course, again, they're going to be the ones that most likely are not to reproduce and survive. But the heterozygotes in this population for sickle cell disease, they are most likely not going to have severe medical problems from having sickle cell trait, and it conjunctively also protects them from malaria. So again, unsurprising that this is most common where malaria is most problematic, which is in parts of Africa. All right, so each of the homozygotes, therefore, then is selected against, because in this environment, HbA, which is normal, they're going to die due to the malarial infection. Oppositely, the ones that have sickle cell disease that have HbS as their genotype, they're going to die to the sickle cell disease. But the genotype that has one of each of those alleles, HbA and HbS, they're a carrier for sickle cell disease. They're going to live due to protection from the HbS gene. It protects them from getting malaria. And then the HbA protects them from getting sickle cell disease. All right. Next, 5.2 is microevolution. So we'll start by defining what microevolution is. Microevolution is small, measurable evolutionary changes within a population from generation to generations. So something I want to make sure that I reiterate is that individuals do not evolve. So instead, populations evolve. And that's one of the common misconceptions that people have that haven't taken a biology classes. They think evolution is occurring, within an individual. That's not the case. So we're not Pokemon. We're not going to evolve and just change. Our genotype that we have once we were conceived, sperm and egg, sperm and egg fuse, and that's what we get. We got those genes from our parents. It doesn't change after that, because after the zygote is formed, mitosis continues until we become adults. So all of our cells have the same genotype, same genome. So individuals do not evolve, rather populations evolve. So the members of a single species occupying a particular area at a given time that reproduce together is a population, and that is what evolves over time. As evolution occurs, genetic and phenotypic changes occur within the population. So all in all, a population has lots of phenotypes and genotypes for the same genes, and we call this the gene pool. All right, so essentially, the gene pool would be if let's say in the group of, let's say that there's 10 of us in this classroom, and we all have 2 genes for eye color. Well, if we took a gene pool, if we're discussing the gene pool, then we would be discussing 20 alleles because we have 2 alleles for every single trait, if it's a monogenic trait, of course. So we could get more complicated, talk about genes that are, or traits that are controlled by polygenic we can talk about polygenic traits as well. But we're going to stick with discussing monogenic for simplicity. Okay, so again, by definition, the gene pool is the various alleles at all the gene loci in all individuals of a population. Now, these the data collected from the gene pool is usually described in terms of genotype and allele frequencies. The genotype frequency, by definition, is the percentage of a population that has a particular genotype. The allele frequency is how much of a particular allele is represented in the gene pool. We'll elaborate. So, now, from the genotype frequencies, you can calculate allele frequencies of a population. So let's do an example. Moth colors. They are controlled by a single gene, meaning it is monogenic and therefore it has only 2 alleles that are possible. As a result, the moths can be either light peppered moths or they can be really dark moth colored. So in this scenario, we're going to use the letters big D to represent the dark color and the lowercase little d to represent the light color. So that means dark is dominant over the light. The genotypes that are possible are big D, which is homozygous dominant, big D, which is our heterozygote, and little d, which is homozygous recessive. So, for example, let's say that the frequency of genotypes in the population are listed here. So this is saying if you were to convert these to percentages, the way you convert decimals to percentages is by moving the decimal place over 2 spaces. So this would be 4%. Likewise, 0.32, I already moved the decimal place over 2 times, that's 32%. And little d is 0.64, that's going to equal 64%. Right, so we moved it over 2 times. So, but for what we're doing right now, we want to keep them in decimal form. I just wanted to show y'all how to get percentages, because how we defined it previously is percentage of a population. Saying that there is 4% of the population is the same as saying there's 0.04 genotype frequency. All right, because they mean the same thing. You convert them decimals to percentages. So anyway, these are the values for the frequency of genotypes respectively. We have 0.04 or 4% that are homozygous dominant, 32% or 0.32 that are heterozygous, and then 0.64 which are homozygous recessive. So we could calculate the allele frequencies by adding together 0.04, which stems from here, plus half of 0.32. Half of 0.32, if you do the math, is 0.16. So if we add 0.04 plus 0.16 together, we get 0.2, or 20% of the alleles in this population are big D. We could do the same thing to calculate the allele frequency based on genotype frequencies over here. We would take half of 32 again, which is 0.16, and then the 0.64. And if we add those together, we get 0.8. So 80% of the alleles in this entire population are little d. And then I want you to also notice, if you add together 0.2 plus 8, or excuse me, 0.2 plus 0.8, what do you get? 1. And 1 is equal to 100%. Okay? So it started off, it told us the genotype frequencies, and then we were able to calculate the allele frequencies of the population given that information. So let's look at this in a genetic context. So let's assume in this population of moths there is random mating. There's no picking and choosing. We can use allele frequencies to calculate the ratios of genotypes in the next generation using a Punnett square as well. So here's our first generation, F1. There's 3 genotype possibilities: big D, big D, big D, little d, and little d, little d. These are the genotype frequencies. They're given, 0.04, 0.32, and 0.64. Then the next thing we did in the previous slide was to calculate the allele frequencies from the genotype frequencies, which we got 0.2 and 0.8 respectively for big D and little d. So now in the F2 generation then, we can calculate the ratio of genotypes in the next generation by putting the male and female sperm and eggs on the top and bottom, just like we did before way back in, I believe, chapter 10, 11, where we discussed Punnett squares. So 0.2 big D, 0.8 little d. Same thing over here, 0.2 and 0.8. And then we would do just like we did with our Punnett squares. We would drag where they intersect. So mathematically, what we would be doing is multiplying. So 0.2 times 0.2 is 0.04. And that would be for big D in the next generation. Moving to the next one, 0.2 times 0.8 is 0.16, and that would be for heterozygotes. We do the same thing here. 0.2 times 0.8 gives 0.16. And again, 0.8 times 0.8 gives us 0.64. Okay, so these Punnett squares are different than the ones that we did in chapter 10. The old Punnett squares that we did helped us to determine potential genotypes and phenotypes of offspring and their probabilities of occurring for individuals, between 2 individuals. These Punnett squares are going to use the allele frequencies of the population to determine the genotype frequencies of the next generation. So I'm going to back up. That's what we did in this example. We took the allele frequencies, which were here and here, and we used those to calculate the genotype frequencies in the next generation. Because remember, all these boxes, they tell us something about the next generation. All right, and so this is telling us there's 0.04, or remember that's equal to 4%, will be big D, big D. 0.32, because I added together 0.16 plus 0.16, which is 32%, will be heterozygotes. And then 0.64, which is 64%, will be little d, little d. Okay, so these Punnett squares use the allele frequencies of the previous generation to tell us what we should predict in the next generation. Notice that the genotype frequencies are the same in the F1 and the F2 generation. So in the F1 generation, we saw 0.04, 0.32, and 0.64. We saw that in the next generation, 0.04. If we add those together, we got 0.32 and 0.64. So what that tells us then is sexual reproduction alone does not bring about a change in the allele or the genotype frequencies. The dominant allele here did not increase. Likewise, the dominance does not cause the recessive allele to just deplete. So what we know about populations is that, in fact, the allele frequencies and genotype frequencies do change over time. This is telling us then, therefore, that just sexual reproduction by itself does not produce these changes. There's some other external factors that do. Now, we'll get to that more in just a moment. Another formula that is utilized to show this mathematical data was created by Hardy and Weinberg. So we call this the Hardy-Weinberg formula. But these two individuals used this binomial equation to calculate genotypic and allele frequencies of a population. The formula is listed here. P2, or P squared, plus 2PQ plus Q squared equals 1. Now, in this formula, P represents the frequency of the dominant allele, and Q represents that of the frequency of the recessive allele. So this here would represent the frequencies of a homozygous dominant. This one would represent the frequencies of a homozygous recessive, and this one would represent, wait, I'm sorry, I said homozygous recessive. This one would represent the frequency of a heterozygote, because it has one of each. And then this one, Q squared, would represent the frequencies of a homozygous recessive. And it's all equal to 1, because 1, remember, is equal to 100%. You can't have more than 100%. Okay, so all in all, in our example, F1 generation, we saw that they had 3 different genotypes and their frequencies were listed below. We calculated the allele frequencies in that generation, and then we used a Punnett square to calculate the genotype frequencies for the next generation. So at the end, we said our genotype frequencies in the next generation is 0.04 for big D, 0.32 for a heterozygote, and 0.64 for a little d. And all of that is going to equal to 1, or 100% of the population. And if you were to insert these numerical values into our equation, P squared would be the frequency of big D, which was 0.2. And if we square it, that gives us 0.4, of course. 2PQ would be the frequency of the big D, little d genotype. So if you were to multiply 2 times 0.2, which was the allele frequency for big D, and 0.8, multiply it then by 0.8 for the allele frequency for the little d, you would be multiplying 0.2 times 0.8 times 2. That gives you 0.32. And then Q squared is the frequency of the homozygous recessive genotype, which would be 0.8 squared. And that gives us 64 in total, 100%. So that is Hardy-Weinberg's calculations. So the Hardy-Weinberg principle states that an equilibrium of allele frequencies in the gene pool will remain in equilibrium as long as 5 conditions are met. No mutations, no gene flow, no genetic drift, there's random mating, and there's no selection. These conditions are rarely, if ever, met. So who cares, right? Why did we even come up with this if this isn't even how the world works? Well, there's a reason. It gives us a basis for knowing whether or not evolution is happening or not. Because, again, in the Hardy-Weinberg principles, there was no change in the allele and genotype frequencies from generation to generation. Again, that's not how things really happen, because we do have changes there, because none of these characteristics are ever met. We know there's new mutations that have to happen because DNA polymerase isn't perfect. There usually is some gene flow. Most of the populations don't mate randomly. We pick and choose who we mate with because we pick and choose who we like or have the biggest muscles, or they're the fittest. Again, what that means for every species would be different. But again, there's typically not going to be populations that just mate randomly. Maybe it's like plant species. They can't really control it as much. But no genetic drift, no selection. These things don't typically happen in a population, but it gives us a basis of understanding the factors that cause evolution, and allows us to detect evolution by noting how much deviation there is in reality from Hardy-Weinberg's hypothetical equilibrium. So we can compare Hardy-Weinberg's principles to the true data of a population over time to see how fast evolution is occurring. So let's go back to industrial melanism, which we mentioned previously, but we didn't really go into details. So there's an increase in frequency of a dark phenotype in moth populations due to pollution. So before industry was really in full swing, maybe a couple hundred years ago, there wasn't a whole lot of industry. There wasn't a whole lot of smokestacks, and there wasn't a whole lot of buildings that put out or emitted byproducts and waste. So during this time, trees tended to be a lot more lighter in color. And so the light-colored moths would escape detection from their predators like birds, and therefore they were more common in the population. But after the advent of industry, dark-colored moths became more common because the trees started to get darker because soot would be settling onto the bark of trees. And so because the background or the environment where these moths lived became darker, it favored the dark-colored moths. And so the light moths would be detected and eaten by these birds. So that is natural selection that occurred in a very short time frame. There's a huge change in the gene pool frequencies that occurred as this microevolution occurred. So let's look at some graphs. So in the beginning, this pre-industrial revolution time period, there was two types of moths, the black and the white peppered moths, and the trees were relatively light in color. So obviously this one here was easier to be detected, and it was the one that was eaten more frequently. So we saw a large population of our moths were going to be the light peppered colored moths, and a relatively low number of the population would be dark. Now, over time, as the post-industrial revolution era creeped up, the trees would become darker in color, and therefore, this time, our lighter moths are able to be seen more as our environment changed. And so what we saw over time as the population numbers of our black moths increased, while the population numbers of our white peppered moth severely decreased. So, of course, along with that, the phenotype frequencies did change, but so did the genotype and allele frequencies as well, because the alleles control the genotype and thus phenotype frequencies. Okay, so now let's discuss the causes of microevolution. Any condition that deviates from the list of conditions for allelic equilibrium causes evolutionary change. So anything that causes a change in the number of, or the change in the allele frequencies is something that causes microevolution. So this is the 5 things that our textbook discusses: genetic mutations, gene flow, non-random mating, genetic drift, and natural selection. Okay, so genetic mutations was the first one. This is our ultimate source for allelic differences, just random mutations that happen to be beneficial or not beneficial. But without mutations, there would be no new variations among the members of our population for natural selection to act on. There is an adaptive value of mutation that depends on current conditions. So in this example here, freckles. So freckles doesn't necessarily benefit or disbenefit an individual. So even though having freckles is a dominant trait, you don't see much of a change from generation to generation in the prevalence of freckles because it's not a harmful mutation and it's not beneficial necessarily either. Next, gene flow. Gene flow is also referred to as gene migration. So if you think about those two terms, flow and migration, there's movement. And that's exactly what this is. There's a movement of alleles among populations by migration of breeding individuals. And this can increase variation within a population by introducing new alleles from another population. So certain species, I think most people tend to think of birds whenever they talk about migrating animals. But if they are typically living in, we have two populations, one that lives in the south and one that lives in the north, as they migrate and intermingle, they can introduce new alleles into those populations. Continued gene flow reduces differences among populations then. Because they're intermingling, two separate populations can become more and more connected and more similar. And this can prevent speciation, because speciation occurs when when a population two populations become less and less similar due to, maybe it's a geographical barrier or some other kind of barrier that prevents them from intermingling and breeding together, and that would cause speciation. So this gene flow can be utilized to prevent speciation. So here's a great example. So throughout the United States here, there are several different subspecies of snakes. So in the light blue region, you'll see this snake. In this region, you'll see this subspecies of snake, and so on and so forth. So all of these different snakes are capable of breeding together. So they're all the same species, they're just subspecies because they tend to look different based on their geographical location. The next condition is non-random mating. So non-random mating means that we are choosing mates according to what we like or what we're attracted to as an individual. So the selection of a mate is according to genotype or phenotype, and it's not just due to chance. So just humans, we do that. We have non-random mating. We don't mate at random. We mate with individuals that we are attracted to that have good personality that melds with ours. Assortative mating is where we tend to mate with individuals with the same phenotype. So we'll see that in species as well. So in this scenario, our homozygotes increased in frequency. Again, sexual selection, we discussed that as well. Where we favor characteristics that increase the likelihood of obtaining mates. Genetic drift, this is where we have changes in the allele frequencies due to chance. So these allele frequencies will then drift, that's why it's called genetic drift, over time. Depending on which members die, survive, or reproduce. This is more likely to occur in small populations. We're more likely to lose rare alleles. And there's two types of genetic drift, the bottleneck effect and the founder effect. Either way, what happens in these two types of genetic drift is we start off with a relatively larger population, and something happens to create a smaller population. So here we see several frogs. There's 10 of them. 9 are green and 1 is brown. So that means 10% of the population is brown, 90% green. Let's say a natural disaster kills 5 of these green frogs, and that's at random. It's just a natural catastrophe like a flood or a hurricane or a tsunami or an earthquake, etc. So due to this natural disaster, 5 frogs died. And that resulted in a population of 4 frogs that are green and 1 that's brown. That changed our gene pool. It changed our genotype frequencies. It changed the allele frequencies. So 20% of the population in this case would be brown and 80% would be green. So the bottleneck effect is where this scenario occurs due to a catastrophe. So in this scenario, species suffer a near extinction, and only a few survivors go on to produce the next generation. So, for example, cheetahs. The cheetahs are extremely similar to one another due to the bottleneck effect, where they had a huge reduction in their population sizes. Oftentimes, infertility is a problem due to inbreeding in these populations because they're so similar. All right, so here's an illustration of the bottleneck effect. So in the original populations, our gene pool is going to have 3,800 alleles. And these alleles are going to be represented by these marbles. So let's look inside this bottle here. So 53% of these little marbles are blue. 26 are green, 13% are red, and yellow, there's 8% of them that are yellow. So to represent the bottleneck effect, if we were to take this bottle, turn it upside down, and only allow a handful of marbles to come out, and then we were to count up all of the different colored marbles, we would see there are 44, and this is hypothetical, of course. There would be overall there would be a great reduction. You see, there's now a great reduction in the number of marbles in the beaker here versus what was in the jar or in the bottle. So here we have 44% of these marbles are green, 45% are blue, 11% are red, and there are no yellow marbles. So we lost a whole phenotype in this bottleneck effect. So it's called a bottleneck because whenever we turn this bottle upside down, the neck of the bottle is so small, it only allows a small number of marbles to pass through. All right, so you can apply this concept to a population. We have a large population of frogs or cheetahs or whatever the case is, and a catastrophe happens that is going to wipe out a huge number of the population, and that's going to result in having a reduction in variation of the population, and that is going to set that population up for potential extinction, because the less variability in a population, the more likely that population is not going to survive. Okay, next we said the other type of drift is the founder effect. This is where rare alleles occur at a higher frequency in a population, isolated from the general population. So alleles carried by founders are dictated by chance alone. So this is where, again, a population, think of what is a founder? They found something, they begin something, they start it. This is where a population will leave the main population and become isolated. They founded a new population, and they are only going to interact with each other. So the Amish, for example, 1 in 14 carries the recessive allele for an unusual form of dwarfism, compared to 1 in 1,000 in most other human populations. So this shows you rare alleles occur at a higher frequency in that case. Just like, again, in the frogs, when we have a large population that was greatly reduced, 10% was the population of brown frogs at first. After we reduced the population size due to chance, again, it was due to chance which ones died. It just happened to be 5 green ones. Then now the population of frogs has 20% of the number of brown. There's still only one frog that's brown, but he represents 20% of the population due to the drastic change in the population numbers. Okay. That is chapter 15. You guys, hope y'all enjoyed that narration. Let me know if you have any questions.